conducted this work with the assistance of two interns, Louise Giuseppe and Melanie Hutchinson. Louise is just finishing up her undergraduate degree in biology, focusing on marine algae, so she was very much useful in this project. And Melanie has just finished her first semester as a graduate student in zoology. And she focused on the invertebrates, general invertebrates, which was really nice. It gave me the opportunity to focus on corals. We also were joined and greatly assisted by various people from the DAR, and we could not have completed this project, we wouldn't have conceived the project without them. So the project was conducted within the context of a larger stud project which is being run with a multi-agency called the Offshore Islet Restoration Committee, or ORIC, which has mostly been focused on terrestrial projects and birds, but it also has an interest in conducting baseline surveys in the marine environment. And some of these have already been done for 11 of the 16 islets that have been designated by the ORC for investigation throughout the main Hawaiian Islands. And these projects, previous projects, may be still ongoing, I'm not sure, were surveys done by DAR and NOAA people. Of the 61 total islets, 44 of them lie in the vicinity of from Oahu to Maui, where we focused. Our objectives and their objectives in conducting these is to conduct rapid assessment surveys. We did it for 2 to 3 islets off of each of Oahu, Molokai, Maui, and Lanai to document unique and sensitive habitats, newer keystone species, species richness and abundance, and introduced or invasive species that may be present. And we evaluated these in terms of the potential or existing threats or disturbance from shore-based human impacts, resource use, or introduced or invasive species. The idea being that islets being somewhat more remote or less visited may be the last best refuge for a more or less pristine marine reef environment. Well, the 10 islands that we finally worked at were almost the same ones that we originally chose in the proposal. 2 of them off of South Lanai, Molokini, and 2 islets off the northeast coast of West Maui. 3 islets in the vicinity of the Kalaupapa National Historic Park. Originally, we were going to do islets over here on the east end, but the logistics turned out to be impossible during the weather conditions that we had. And finally, 2 islets, Kaiohikaipu off of East Oahu, and eventually we settled on the island of Kapapa, the outside of the island of Kapapa. All these had rather diverse and different habitats, and we did come up with some surprises because we did have quite variation in results among them. To do this, I wanted to do as much as possible to stick with protocols that have been used by NOAA and DAR in their projects, but we were constrained by the fact we had 4 people due to boat limitations and also budget and personnel, whereas these NOAA DAR projects are rather big things. They go out in 3 boats, 3 teams, and 8 people at least to conduct it. So we made some modifications of their methods, which involved having 2 dives at each site, one which was a general reconnaissance where we all swam around and identified or collected those things, identified what we could and collected some of what we couldn't. And 2 additional, the second dive was to do algal, coral, and fish transects using our revised protocol. We marked the vicinity of our transects and reconnaissance surveys with falcon buoys, took GPS coordinates so these can be reproduced in the future. And this gives us georeference data on habitat characteristics, fish species, and biomass, and benthic coverage, which we determined using photo images and coral point count analysis. And we also visually identified and photographed invertebrates and macroalgae of interest. And what we couldn't identify in the field, as I said, we collected and brought back to the laboratory. Well, just I talked at more length about the method that we used last HCRI presentation time, so I won't go into any detail. But basically, we would use the photographic quadrats to get an image which we could bring back to the laboratory. And using very convenient part of the coral point count analysis to outline the areas of each individual coral, we could not only get the size class analysis, which is the objective of the DAR NOAA method, but also end up with areal coverage estimates. And for the algae, again, a smaller quadrat was photographed, and Louise analyzed these using random points on a point intercept analysis to get an idea of the composition of the algal communities. And the fish invertebrates fish transects were done using the DAR protocol of 3 25-meter long transects by 4 meter wide belts, and all fishes were recorded by size and species. And Melanie, acting as the partner to the person who did the fish counts, also recorded the invertebrates that could be identified on a 1 meter wide transect for the 3 transects, and at the end or the beginning collected a standard size coral head for associated invertebrates. Well, these are the sites that we did, and the two on South Maui were the most accessible. This one's Pu'upei is right around the corner from Manele Bay, Manele Harbor, and a very similar site only about 5 kilometers away at Po'opo'o. These are both very similar in location, exposure to wave and wind, and both of them being considered sheltered sites. On Maui, we did 3 sites. These two, Molokini, probably all of us are familiar with, and Molokini is probably one of the most dived and scuba or snorkeled places in the entire Hawaiian Islands, with a massive number of people going out there every day, but they stay inside the crater. Our surveys were conducted on the outside of the crater, which is an entirely different environment, almost unique in Hawaii, I think. The two on the North Shore are quite inaccessible in terms of the normal trade wind conditions. It's really tough to get out there, or more precisely, it's hard to get back. And we were really lucky in getting some, excuse me, having some really good weather while we were there. So it was a great opportunity to visit a place which has not been studied, I think, at all before. Off of Molokai, with the assistance of Eric Brown and using the facilities of the National Historic Park there, we were able to get out to Okala and Mokapu around the peninsula from the Kalapapa colony. And we were going to go, as I said, originally over here to the east end. That proved impossible. We had settled on going to Huelo, which is the third islet within or near the park boundaries, but we couldn't get out there on the third day. It was so rough. So we did a very small, low-lying islet that's right off of Kalapapa Harbor, and that's called Namoku.